Hey everybody, so this video is going to be part of the plant unit, so this will be the plant lab. So I'm going to go over all the things that we would have done in lab, and this is the things that you'll be responsible for that portion of the lab exam. So this is a little different video setup than what I'm used to in my office, so forgive me if I'm toggling back and forth and may get lost because I'm trying to figure it all out. So this is my first go round with this particular software. So anyway, so I'm going to get my PowerPoint loaded up and we will talk about some of the structures that you need to know. Okay, so we'll toggle me off and go to my PowerPoint, which is somewhere. There we go. Okay, let's see if I can make this full screen-ish. Okay, that should work. All right, so these are going to be the structures. So I'm going to start with the mosses and the ferns, and then we'll do basically the gymnosperms and then the angiosperms. So if you remember from lecture and what you learned about in lecture, the plant world is divided into the ones that don't have a vasculature and then the ones that do have a vasculature. So the mosses, no vasculature. They're small enough for things to move through osmosis and diffusion. Ferns are your first vascular plants. And then you have the gymnosperms, which are like pine trees, and they have vasculature, so they can be large. They have the ability to move things all the way from the ground to the top of the tree and back down. And then the angiosperms, which are your flowering plants, which includes flowers and also large trees like oaks and magnolias and things like that. All right, so one thing I'm not going to do in this video, because I want you to kind of go back and fill in those gaps. So as you're going along, is paying attention to which of these parts of the plant is the 2N versus the 1N part. So the haploid versus the diploid. So I do need you to kind of go back and look at your book and look at the lecture notes and make sure you fill in those kind of gaps. All right, so this is going to be the moss. And so if you look at a moss, you'll see that on what it looks like the same plant, you have both of the alternation of generations. Remember, plants have a sporophyte and a gametophyte generation. Okay, they alternate between the two. So in the moss, this is the gametophyte generation. So everything below and then everything above is the sporophyte. So the parts that you're going to need to know, in this PowerPoint, I've been trying to label everything that you need to know, so you really can use this as the guide for what you need to know for the lab test. So in the gametophyte generation, you have the leaves. Okay, so these would be leaves. And this is where the male and female portions will be. On some of these, there's a male portion, and on the other one, there'll be a female portion. And my next slide will show you that. The leaves are held on by a stem. And under the ground, you have those rhizoids, which are not actually roots, but they do allow for things to be absorbed from the ground. But they're not true roots because in the case of a rhizoid, these can spread underneath the ground and allow another plant to originate. So it's kind of like an underground way for the plants to spread. So coming from this portion here, you have this long stem-looking structure called a seta. And on top of that, you have the capsule. And this is where the spores are. So if you go back and look at your moss life cycle, this would be a good time to get your book out and look at the moss life cycle and see what's going on in there. This is where the spores are going to be released. And at the tip, there's an opening that will basically allow it to burst open called an operculum. So that's where the spores will be released, and then the life cycle will start over again. So again, take this and then also compare this to the lecture material on the alternation of generations. Now, in lab, we would take a microscope and we would actually look at these sex portions of the moss. And so there's a male portion and a female portion. So this is what it would look like, the green, green portion of the plant. If we look at a microscope, you're actually going to see an ovary-like structure with an egg in it. Okay, so this is going to be the female portion. And in the male portion, if we looked at the microscope, you would see these structures that would contain the sperm. Okay, so the female has the egg, the male has the sperm. So we have some special names for those, Antheridia and Archegonia. So if you remember, I actually reversed these so that you would be paying attention and not just trying to memorize it, actually paying attention to what you're seeing. So the Antheridia, that's going to be the male portion. Okay, you can see these have the sperm cells, and the archegonia, okay, that's going to be the female section, or the female part, and you see this area with the egg inside. Okay, so the antheridia is male and the archegonia is female. Okay, so those are things that we would have looked at on a microscope in class. So this is the next group of plants. So we're going to start with the ferns, which have vasculature. So they're bigger, so they have to have a way to move substances larger, further distances throughout the plant. Again, ferns have alternation of generation, so I'm going to leave this to you to go look in your book, look in your notes, and tell me if this is the 2N or the 1N part of the life cycle. So this is a gametophyte, the fern gametophyte, and it has this heart-shaped structure that's called a thallus. Okay, so that's another thing that you'll be able to identify on the lab test. And this is where sperm and egg are actually going to be able to come together and start the next generation. Okay, so this is the gametophyte generation. Now the sporophyte generation, this is what we think of when we think of a fern. We typically don't really see that thallus. That's sometimes just underneath the ground. It's kind of small and we don't actually see it. The fern is what most of us recognize as the sporophyte the sporophyte generation is what we actually recognize as a fern. So it has roots, okay, and these are connected to a rhizome, so this allows that underground spread. Okay, so it's kind of like a stem. It's kind of like you planted your tree underground and the leaves were growing out of the ground. So the rhizome is kind of like a stem, if you want to think about it that way, with roots going into the ground. So this is the fiddlehead. It's a little moved, should be right here. This is when these fern leaves start to develop, they basically have to unroll themselves, and they unroll themselves out until they become these leaves, and we call that a fiddlehead because it looks like the end of a fiddle. So in the fern, from all the way here, all the way to the tip, that whole thing is a leaf, okay? Not these little things, these little leaflets, okay? But this whole thing is a leaf, okay? And then on these little bitty leaflets, if you turn it underneath and look on the underside, you're going to see where the spores are located. So these are called the sorus. So multiple, I've used plural, sori, one is sorus, okay? So this is where the spores are. So again, take some time. This would be a good time to pause, go to your book, look at alternation of generation and see what's going on. So what is the reason for the sorus, what's being held there, and what's going to be released. So this is where those fern spores will be released so that the new generation can begin. So again, this would be a good time to go in and write in here, is this 1N or 2N, haploid or diploid? All right, so the next two things we're going to cover are your gymnosperms and angiosperms. So I've got this slide you've seen before. So the gymnosperms are your pine trees, okay, and your angiosperms are your flowering plants, which includes roses and lilies, but also dogwoods and oaks and peach trees and pear trees, anything that has a flower. And these are all vascular. Okay, and for these plants, these are the ones that what we typically recognize as the plant. This is going to be that sporophyte 2N generation. Okay, and then we're going to cover the flowering portion is going to be the 1N or the haploid version, and then where the sperm and egg are made on the pine tree, what we think of as cones, that's going to be the 1N generation. So let's go through each one of these and point out some structures for you to be tested on. So in the gymnosperm, if you've ever noticed on a pine tree, there's two sets of cones. A lot of people have seen these and they just assume that that's just an underdeveloped version of that, but it's not. There's actually male and female cones. All right. So the male cones makes pollen, which is essentially sperm, and the female ovulate cones, they make the eggs. So pine cones are female and these pollen cones are male. So if you're watching this the last couple of days, my car's been covered in yellow. That is the pine pollen. So the male pollen cones right now are really releasing all their pollen that's drifting through the air, hoping to find a female ovulate cone so fertilization can take place, and then you'll have some development of seeds. And then the seeds can fall, and that will help grow a new pine tree. Okay. So let's look at an actual pine cone. So this is a pine cone where the pollen has reached the female ovulate cone and fertilization has occurred, and we have the production of a seed. Now, gymnosperm means naked seed. All right, so these seeds aren't covered in like a fleshy fruit. Like think about an apple or a peach or anything that has that's coated. These are just naked. They don't have any kind of protective coating around them. So gymno means naked seed. And so these seeds are found within the pine cone, and these little kind of protective structures are called scale. So if you take a pine cone, a lot of times when you first see them, they're really, really tight, and then they start to mature, and the pine cones start to open up, and then the seeds can fall out. And then they can be dispersed by animals or by the wind, or sometimes in the case of some pine trees, it takes fire, it takes heat to cause those pine cones to open up and the seeds to fall. All right, so these would be structures to know for the pine. So that takes us to the angiosperms, which are your flowering plants. And again, this is anything from what you might consider a weed to a rose, to an oak tree, to a dogwood, to anything like that. So some basic parts of the flower, that's what I want to focus on right now, is the flower. If you look at the flower of an angiosperm, they all have some basic things in common. The flowers have both male and female parts on them. So this is going to be the male part. And it's called the stamens. And so each stamen is made up of these filaments. And on top of the filament is an anther. And the anther is where the pollen is. Okay, so these are the male structures that have the pollen. And then these typically surround a central female section called a carpel. Okay, and the carpel has an ovary. It's called here, basically this round protective structure. And then inside here you have the ovule, which is the egg. So the female has the egg. And leading from this ovary, you have this tube called a style, and the tip of the female section is called a stigma. So the pollen would land here and form a pollen tube in order to get the DNA down to the egg for fertilization to occur. So male portion and female portion. The flowers, which we call the petals, are also the corolla. This is what we think of when we look at a flower. These are usually decorative. They're brightly colored. This is attractive to insects or birds so that they'll come to the flower, so that then if it needs pollination, whatever animal, bat, butterfly, bee, the pollen will come off the anther, and then they'll deliver it to the next flower. It'll hit the stamen, and fertilization will occur. They don't all work that way. Sometimes it's just wind, but sometimes you do need a pollinator like a butterfly or a bee. So a lot of times these flowers are really colorful and attractive. Underneath is this green stuff. This is called the sepals or the calyx. So if you've ever thought about a rose bud, you may have seen like a green, like if you pick up roses and they're kind of closed and it's surrounded in like a green shell. That's the sepal or the calyx. And you can use either term. I'll accept either. So that opens up and then the flower is inside there. So a lot of times if you see a flower before it's opened, it's surrounded by this calyx. And then that calyx kind of forms the base, and then the flower, will basically appear above that. Okay, so think about that when you think about like a bud. This is what you're seeing before the flower opens. So if you remember, we have this model in our lab. So you also, in your Blackboard, there's a packet that has a bunch of this stuff with images or diagrams that are labeled. So I'm putting these side by side so that you can understand a visual of a diagram and the model at the same time. So I may use either or of these images on my exam, so make sure you study both. But on the model, this will be the calyx. You can see this is going to be where the petals, the corolla, come from. Okay, this is going to be the male section with the filament and the anther. So you got the filament and the anther. That's where the pollen is. This is going to be the female section. So you have the ovary, you have the egg or the ovule, and here's your pollen tube coming down to fertilize the egg. Okay, so same thing over here. You can see exactly the same thing on the diagram that you see in the model. All right, some other things about plants is you have to know, because they have vasculature, they have more structure. So plants have stems and branches and leaves and roots. So just a few things to talk about in regards to that. So this would be the stem. And if it's just a single plant, the central thing coming out of the ground would be the stem. And then like on a tree, you would start to see branches. You would call that the trunk. And then little branchings coming off. But if it's just a single plant like a zinnia or something like that, then you just have a stem with leaves coming off. Okay, and wherever a leaf or a branch comes off, we call that a node. Okay, and then between two nodes, so here's a node and here's a node, or right here actually. So all this area between the node is called the internode. Okay, so if you go back and think about the lecture material, this is where you have those meristems. This is where the mitotic growth is occurring. So it grows node to node to node. So the nodes are the growth plates. Think about how bone grows. Have you ever heard about how bone grows at growth plates? And it's kind of the same thing. It's growing at the node, and so the internode is those regions between the node. So 3 things you can identify on this particular slide would be a stem, a node, and internode. We have roots. All roots are not the same. Different plants have different types of roots. This is called a fibrous root system. You may, if it's ever been your job to go weed your yard and you're trying to pull up, especially grasses that are not the grass that you want. Maybe you've got Bermuda, now you've got fescue growing in your yard. And you're pulling up and you've got this big old clump of roots that come out with it and brings all the soil with it. That would be a fibrous root. Carrots have a long taproot. So what we actually eat is actually the root. So it's usually a long single root with some small hairs coming off of it for absorption of nutrients and water from the soil. In Alabama, there's a plant called Queen Anne's lace. It's got a really pretty white flower you see in the summertime. It has a long taproot, and if you pull one up, you'll notice it looks just like a carrot. It actually is. It's a wild carrot. The taproot is white. So we do have a wild version of carrot here in Alabama. Prop roots, a lot of times, this is what you see with corn. So it almost looks like it's, so you'll see the main stalk of the corn or the stem of the corn above the ground, and you've got the roots coming off at an angle above the ground and also those above the ground. Makes them very, very stable. So it allows them not to be blown over in the wind. Mangroves also have prop roots. So if you want to Google mangrove and have some really good ideas of what a prop root looks like, that's a great example. And then adventitious roots, they like to take advantage of whatever they can. And so a lot of vines have these adventitious roots. It allows them to grow along up a tree and absorb water from their surroundings on the bark of the tree as they grow. So adventitious roots. So these 4 things, you may see a picture of one of these 4 things, and I'll say name the type of root. One of these 4. All right, so we also, if you have in these plants, these angiosperms, you've got your flower, you've got your stem, you've got your roots, and you got your leaves. So let's talk a little bit about the roots next. So again, some of this was in your lecture material, so this is just kind of hitting the highlights of what we would have identified in the lab. You probably have seen this model and we're wondering what it was. It's a root. It's called a root apical meristem, and so this is what this is. We would have looked at that in the lab using the microscopes. So just going to have you identify 4 parts of the root. So you have the root cap. Okay, so this is going to be this section. This section. It's a protection for the root. So as the root is burrowing through the soil, it protects those more delicate structures underneath. Okay. Then you have the protoderm. So it's going to be, think about that as being the epidermis, basically. It's the outside of the root. So the root actually has to have structure. So this is the skin, if you will, of the root. All right. And then between the skin and the internal portion, you have what's called the red arrow should be the ground meristem, sorry. These are the same. This is the ground meristem. So this will be this yellow portion here. So think about the root having an outside, a middle, and an inside. So the ground meristem is the middle portion. And you can start to see some tubes and some vascular structures in here. And then you have the procambium. Okay, this is the inside, kind of the most innermost portion of the root. All right, so here. So again, the functions, this lecture is just to show you where they are and how to identify them, what they do. You can see in the lecture and also in your textbook. All right, so you with a plant, you have a flower, you have a stem, you have roots, you have leaves. So this is something that you're going to want to pay attention to when you're doing your tree identification. When you're trying to figure out a tree and what it is, one of the things you look at are its leaves. And for each leaf, you're going to look at the type of leaf it is, how it's arranged, and what its margins look like. Okay, so this is, on this slide, we have leaf types. Okay, and so a simple leaf means here's your stem or your branch, and one leaf comes off of it. Okay, so just stem, one leaf. A compound leaf, here's your stem again, and what you see are these leaflets. So here's 1, 2, 3, 4, 5. This whole thing is the leaf. Each one of these is a leaflet. That's a compound leaf. This would be an example like a hickory or a pecan or a black walnut. So if you were collecting this, you have to collect the whole leaf. Okay? You can also have what's called pinnately compound. That means feather-like. So again, here's your stem or your branch. This whole thing is the leaf. You'd have to collect that whole thing, and each one of these leaflets looks kind of like a feather. Locust trees here look a lot like that. Mimosa trees too. The ones that you see in the summer with the pink fuzz balls on them, those are mimosa trees. They do that. And then this last one is called palmately compound, where again you have a stem, and you have the petiole or the base of the leaf, and then the leaves kind of come off all from the same point, kind of like you're spreading your hand like imagine these being each one like a finger. So a great example locally here would be a buckeye. So the buckeye, like Ohio State, that's a palmately compound leaf. All right, so you also want to pay attention to how the leaves are arranged. When you're doing your leaf identification or tree identification, one of the questions is how are the leaves arranged? Are they alternate, opposite, or whorled? So what that means is you're looking at the stem and you're looking at the nodes. So here, node here, node here, they're on opposite side and there's space between. Those are alternate. If you have a stem with two leaves coming off at the same place across from each other, that's called opposite. And if you have multiple leaves coming along kind of like a circle around it, those are called whorled. All right, so these are the three things you're looking for, trying to figure out if your tree has alternate, opposite, or whorled leaves. One more thing, I didn't make a separate slide for it because I was just going to go back and look at this one. The other thing about leaves, when you're describing your leaves and looking at them, it's going to ask what's the margin. Tell me like the margin of the paper. I'm really missing my pen that I have in my office. And that means what's the edge look like? So does it look toothed? So serrated? Is it smooth? Does it have lobes? Does it have big jagged teeth? Does it have little tiny teeth? So if you look in your tree identification book that's in your blackboard that's from Auburn University, the Extension Service, it gives you examples of what those leaf margins look like. If they're rounded, if they're toothed, if they're jagged, and that kind of thing. So that's what margins are. So what does the edge of the leaf look like? All right, so the last section, when we think about angiosperms, okay, this has to do with the angiosperms, okay? So put a big A here, angiosperms. They're divided into two big groups. monocots and dicots. All right, so when we classify the angiosperms, some of them are monocots, some of them are dicots. So what makes a monocot? What makes a dicot a dicot? And so what you're looking at are these 5 structures: the root, the stem, the leaf, the flower that we just talked about, but also the cotyledon. This is the embryonic leaf. This is what, if you were to open up the seed, that you would see inside the seed. This is what's going to germinate and become the plant. So in a monocot, it has 1 leaf, and in a dicot, it has 2 leaves. Okay, and again, this is lecture material, but that's what you're looking for. Now for us, we're not going to pay so much attention to the cotyledons because what we're going to see mostly are going to be the roots, stems, leaves, and flowers of these plants. So the roots are going to look different. You're going to see a star-shaped center in the dicot. In the stem, you're going to see a more regular bundle of vasculature in the dicot. It's more random in the monocot. The dicots have more advanced leafing. You can see a lot more branching. Monocots are more simple. And the dicot leaves typically have more petals. All right, so we're going to go through each one of these briefly. Starting with the monocots, again, you have 1 cotyledon in the embryonic leaf. In the root, which is here, you have xylem and phloem in a ring formation in the center. In the stem, the xylem and the phloem are kind of randomly dispersed all throughout the stem. The leaves, the veins are parallel; they aren't branched. If you think about a grass leaf, so if you think back to the old days, maybe you were sitting on the playground picking grass and maybe stripping the leaves and ripping them into long strips, like how bamboo looks, those long parallel leaves. And then the flowers, their multiples are always going to be multiples of 3. Okay, so those are some things that monocots have in common. So if you could be in lab, we would look at this under a microscope. So this is a microscopic image of a monocot root. Okay, so make sure you're paying attention if you're looking at a root or a stem. So in this case, again, I'm cutting down. This is what you need to know. In a monocot root, you have a pith. Okay, and so when you look back at your lecture, there's more information about what all that means and what it does. You have a cortex, which is going to be this outer ring of material. Okay, and again, these are angiosperms. That means they have vasculature. So they have xylem, okay, which is for water, and phloem for food. So these big white things are the xylem, and then in between is the phloem. Okay, so it's just xylem, phloem, xylem, phloem, xylem, phloem. All right, this is the stem. Okay, so you have these vascular bundles. So that's a vascular bundle, that's a vascular bundle, that's a vascular bundle. And they're just kind of random. They're kind of randomly dispersed throughout the stem. There's not really a central pith. So there's no central region. It's just all kind of randomly dispersed within that. If we take one of these and blow it up, we see that this is the xylem, okay, and then the phloem is just above that. Okay, so this little section here is the phloem, all right, and it looks like a face. So if you're old like me, you know this is Mr. Bill, and it looks kind of like Mr. Bill. So the phloem would be the hair, and the xylem would be the 2 eyes. Okay. This is another image of the stem. Look familiar? This has been in your lab. It was not a big piece of pizza. This is a monocot stem. You can see the vascular bundles are spread kind of randomly throughout the stem. And again, you have your xylem and your phloem. Okay, so the phloem is going to be the hat or the hair, and the xylem are going to be the eyes. Okay. All right. Two other things two other types of tissues that you may have may remember from lecture are cholenchyma and sclerenchyma, and so these are also labeled here. You'll see the cholenchyma forms this layer just beneath the epidermis, which is the outer layer of the stem. So you have a stem and then a layer of cholenchyma, and then this fibrous sheath or sclerenchyma kind of surrounds the phloem. Okay. So here's a version or a picture of the monocot with the definite parallel leaves, parallel veins inside the leaf, and a flower with multiples of 3 petals. All right, dicots. You may see the word eudicot, just means true dicot, so either one is acceptable. You have two cotyledons. Okay. In the root, this is the root, you see this X-shaped xylem. So the xylem is cross-shaped here. And then you have phloem between the arms. So it's got more structure than what we saw before, a little more advanced. And then with the stem, you have this definite rings of xylem and phloem. So you have xylem and phloem making these outer rings. So this is going to start to be the thing that you see in trees. Okay, so you know how we figure out how old a tree is? We're counting the rings. Because of this particular setup, these are allowed to grow year after year after year, making our woody plants. And so that's what we count. We're looking at these different rings of xylem and phloem to tell us how old the tree is. All right, these are the leaves. Got more branching, more defined, definite branching, more veins. And then multiples are 4 or 5 are going to be your corolla, or your petals. So again, we want to look at the microscopy of that. So I've tried to label it for you. So the star-shaped vascular cylinder, what you have are alternating patches of xylem and phloem. So the phloem is in green, if you can tell that. So phloem, phloem. The xylem arms, so xylem, 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 xylem. Okay. You have an endodermis, so something that basically holds those vascular bundles together. You've got to have, gives it structure. And then you have pericycles and a cortex. The cortex is kind of what's around that, outside the endodermis, kind of gives it more structure. And the pericycles have to do, they're like gates that allow moisture to move in and out. So again, that's lecture material, but they're going to be just around kind of those phloem areas. So so that was a root. So here's the stem. So I'm blown up a section of a stem, and so what you see is a clear epidermis. You can actually see there's a literal layer all the way around of epidermis. You have a central pith. So all of this in blue is all of that. It's all of this. That's all pith, which is parenchymal tissue. Okay. Then you have a cortex all the way around here, just below the epidermis, but outside of these vascular bundles. That's cortex, and that's also parenchymal tissue. And then you have the vascular bundles. So that's a vascular bundle. That's a vascular bundle. That's a vascular bundle with the xylem and the phloem. All right, so let's blow that up a little bit. So here's the vascular bundle. And so you have a bundle cap here. Looks kind of like an acorn. Okay, so think about the bundle cap as being the cap of the acorn. And then you have phloem all in here and xylem toward the center. So those would be the things to identify. So this whole acorn structure would be basically a vascular bundle. The cap will be the bundle cap with the phloem and then the xylem toward the center. And then this is pith. And there's the epidermis, this layer right here. All right, so again, look familiar? This is in the lab. So this is a woody dicot stem. And remember, this is because you have these alternating, these regularly displaced vascular bundles. It allows these things to grow year to year, and they grow in rings. So again, this would be, if you're looking at this particular diagram, this is in that set of diagrams that you've got in Blackboard. These are the phloem rays here and here. This outer layer, this is what we consider the bark. This is the outside of the tree. Okay, and then it's kind of superficial or more to the outside of that is what's called the cork or cork cambium. So the stuff that peels off, when you think about bark peeling off, that's what you're seeing. And then you got this innermost bark. And then this is the part that's, that's growing. I mean, you've got your xylem. Your xylem is growing from year to year, and each one of these layers is a ring of annual growth. And so what you typically see is large spring growth and then kind of compact summer wood. So you typically see 2 layers in each annual ring. So all of this stuff that's listed here, I'll probably have this particular image and use something from these choices over here to ask you to identify. So you can take the same information, and here's your pith, okay? Here is your wood, all right? And here is your bark with your cork cambium, and this is going to be where your phloem rays, okay? And all of this is going to be the xylem in these annual rings. All right, so you can take the information from here and use it over here. All right, so the last thing will be the leaves. And so the dicot leaves, regular branching, so it looks lots of veins going off in branching patterns, much more complex than what you see in the monocot. And the flowers are in multiples of 4 and 5, the corolla, so the petals. Okay. All right. So the last part, since we're talking about angiosperms, that means they make some kind of a fruit. Their seeds are not naked. They're encased in something. There's a lot of different types of fruits. I've narrowed it down just a couple things I'm going to cover for you this semester. So I want you to know what a berry is. And so with a berry, you have a fruit wall that's soft and fleshy, and the inside's usually kind of slimy. All right, so kind of like a grape or a tomato. All right, so these are both examples of berries. But not all berries have a soft outside. There is a group of berries that have a more tough outside, but they're still slimy on the inside. And that's your citrus fruits. Okay, so with your citrus fruits, they're actually called the Hesperidiums. They're really a berry, but they have a tough outer rind. Okay, and so the word I want you to know is exocarp. So lemon peel, orange peel. This is the exocarp, and that's where the orange oil, lemon oil, if you squeeze one, if you ever squeezed an orange lemon, you've seen the stuff squirt out, that's the oil. That nice fragrance that you get of lemon and orange is going to be in that exocarp. All right, just deep to the exocarp, you have that white part. That's called the mesocarp. All right, a lot of times that can be kind of bitter when you're peeling an orange. Sometimes that stays on there and you have to kind of peel that off. It may not taste so great. And then just below there is the endocarp. And this is what makes our slices. So that's the good part you want to eat. Okay, so that's the endocarp. And then inside of each one of these, you have these little carpels. So remember female part, the carpel. And so that's what's going to be filled with these little juice. That's going to be the juice sacs. And then toward the middle, you have the individual seeds. Okay, so there are your words for this particular fruit: exocarp, mesocarp, endocarp. The section, which is called a carpel. The little bubbles that you pop that are filled with juice, called the locules, and the seeds. All right, the next one are called your drupes. All right, so drupes are fruits that have a soft, fleshy part that you eat, usually a really thin skin, and a really firm one single seed in the middle. And it's called a stone. Usually, these are called stony fruits, stone fruits. Peach pit. That's example of what a stony seed looks like. So this is a peach, and these are cherries. So the pit of the cherry is actually its seed. And so the three things: the outside, the soft outside skin, the exocarp. The good part that you eat is the mesocarp. Then that endocarp, that's the outside shell of the seed. The seed's actually inside there. So you may have cut into a peach one time when you found the peach pit, and it looked kind of like an almond. And then if you could open up that almond inside, you would find the seed. And that's literally what almonds look like. Almonds look like peach trees, and so but the fruit's not really good. So they get rid of the fruit, and then what you're left with looks like a peach pit. You open that up, and then what you eat is the almond is the inside. Okay, so the exocarp, so the endocarp, sorry, is the pit of the peach with the seed inside of it. The next group are the pomes, and these are apples. And also pears. So apples There we go, apples and pears. And so what you see from these apples and pears, these pome fruits, is that you have the fruit is going to be basically surrounding the ovary. So this is the remainder of the calyx. So the flower was there, and what you still got that little calyx left. And you can imagine that was the ovary with the little eggs inside and that have been fertilized. And then you get this fleshy stuff develops around it to protect the seeds so that hopefully somebody will eat the seed or eat the fruit, eat the seed, and then carry the seed with it, poop the seed, and then the seed will grow. That's kind of the plan there. All right, so parts of the apple, obviously you have the stalk. The calyx is going to be on this end, the other end, opposite of the stalk. Okay, so on this one, the stalk would have been on this end. So then you have the outside exocarp, middle mesocarp, and then around the ovule, that part that you usually try to cut away, what would you think of as the core? And it's kind of hard, you don't like to eat that. That's the endocarp, and then the seeds are inside of there. Okay, so here's all your words to learn for poems. So apples and pears. And so I may have an actual apple or an actual pear that I've cut into and taken pictures of for your test. And that's it. That's all the slides. So this will be your lab for the plants, and then you'll have a few more short videos covering fungi and tree identification as well. But this one should cover everything we would have done in lab for the plants. I'm going to take it back. Say thank you very much. I know this is kind of a weird time, so I appreciate your time, and let me know if you need any help. And I will see you soon, I hope. Bye.